Now that you've seen the general functions and the anatomy of the basal ganglia, let's take a look at a couple of disorders that are associated with degeneration of different parts or different pathways of the basal ganglia. So I'll start in this video with a little info about Parkinson's disease. So what does it look like? How does it relate to these pathways we've been talking about through and into and out of and within the basal ganglia, and what are a couple ways in which it could be treated? Then I'll move on in the next video and talk about the damage that's associated with Huntington's disease, as well as some ways in which that disease is treated. So I'll just remind you that the basal ganglia includes a lot of different structures within the brain. The ones we're going to be focusing on here will be listed. So let's look back here. And the first thing I want to do is just kind of do a thought experiment. And basically, how would voluntary, it's answering this question, how would voluntary movement be affected by degeneration in these different pathways? So we'll start with the D1 pathway. Remember that that's going to be the D1 receptor is involved in the direct pathway. And so if we're inhibiting that D1 pathway, we are inhibiting an excitatory input into this direct pathway. So if that is no longer excitatory, that means these neurons will no longer be active. If those are no longer active, that means that they no longer are providing any inhibition for these internal segment neurons. So they're going to be able to inhibit the VAVL complex, and that is going to prevent the activation of the voluntary movement. So if we get rid of the D1 pathway, we get rid of the voluntary movements, or at least we decrease them. Okay, so let's move on then, and we will then look at the D2 pathway. What happens if we degenerate that one or get rid of that one? So remember, this D2 pathway is involved in the indirect pathway. So if we get rid of this, now we have basically allowed for these, the caudate to external segment pathways to become active. They're inhibitory, so that's going to inhibit the globus pallidus. So here we have one inhibitory pathway gone, two inhibitory pathways gone. Oops, there we go. And once we do that, you can see hopefully that we now have no more inhibition of the subthalamic nucleus is a big one, and no more inhibition of the internal segment. This means the subthalamic nucleus is available to excite the internal segment. It means that there is at least one missing inhibitory component on the internal segment. So overall, the internal segment is going to be more active. So it's more active, that's going to increase the inhibition of the VAVL complex, which once again, we see inhibition of voluntary movements in the frontal cortex. Okay, and so now we have one more that we're talking about here, and this one is degeneration of this connection between, this connection between the caudate and the globus pallidus external segment. So that's the indirect pathway again. So basically, you're talking about knocking this guy out. If that one is knocked out, now all of a sudden you have increased inhibition here, increased inhibition here between the external segment and internal segment, and the external segment and the subthalamic nucleus. In both of those cases, that is going to increase the inhibition of the internal segment. That will decrease the inhibition of the VAVL complex, and we're going to see increased activity in the frontal cortex related to voluntary movements. All right, so you can see that inhibition of any of these initial steps in this basal ganglia pathway, direct or indirect, we're going to see very pronounced effects on voluntary movements through the frontal cortex. So then here's another question, and this one is related to something very real, which is Parkinson's disease. In this case, what's happening is actually there is a degeneration of both of these pathways. So we have degeneration of the D1 and the D2 pathways. And because of this, we're going to see a lot of different effects. So now all of a sudden, the excitatory input into the direct pathway is gone. So there you go. Inhibition is gone. This is going to increase the activity of the internal segment and increase the inhibition of the VA-VL complex. Similarly, we've got the D2 inputs to the caudate are now gone, and or at least very diminished. This is going to prevent the inhibition of these inhibitory indirect neurons. That is now going to lead to inhibition of this external segment. And so then now we see the activation of internal segment neurons as well as the lack of inhibition. That means these internal segment neurons are quite active at this point. Because of that, they're going to inhibit the VA-VL complex, and that is going to inhibit voluntary movements. Hence, you can tell now, you can see the underlying basis for how Parkinson's disease becomes a hypokinetic disease. This is a disease of diminished movement. Here are some of the manifestations of Parkinson's disease. One may be surprising if you're thinking about this as a hypokinetic disorder, and that's tremors. So you definitely see tremors occurring. This is just sort of increased baseline activity in different muscle groups. However, the others are going to make a little more sense for this hypokinetic disease. In this case, we have rigidity, so that's going to include things like reduced facial expression, as well as slight flexion in a lot of your different major joints. And the others are stooped posture. So basically, again, that's probably related to this rigidity, the stooped posture. You're basically pulled over a bit. And then finally, bradykinesia. And this is maybe the most famous part of Parkinson's is the slow gait. They tend to shuffle. People with Parkinson's, they have slurred speech, and they have a lot of difficulty with fine motor movements. They tend to have trouble getting those movements started as well as actually performing the fine movements. So let's look back to the 1970s, and this is when some evidence was coming out for the etiology of Parkinson's, so basically the basis for Parkinson's. There was a post-mortem study done in which 69 different brains were studied morphologically. And in doing so, they noticed that the dopaminergic neurons in the substantia nigra pars compacta were damaged. So basically, they could look and they could see that the substantia nigra pars compacta was actually smaller, and it was those dopaminergic neurons that were damaged or missing. They also looked at 28 Parkinson's brains biochemically. And when they did that, they actually looked in the corpus striatum and they found reduced concentrations of dopamine there, which is what you would expect if there were fewer dopaminergic neurons releasing dopamine into that area. So there was a pretty straightforward, I mean, it's not a simple study, but pretty straightforward results, both morphological and biochemical. So what can we do to help alleviate some of the symptoms of Parkinson's disease? Maybe try and think of a couple of ways. Remember that the bottom line here is that we're seeing decreased excitation of the frontal cortex in Parkinson's disease. So we're trying to increase that excitation in order to overcome a lot of the deficits that are seen in someone with Parkinson's. One way in which you might be able to do this is to replace the dopamine. And so in this case, you would need to provide dopamine to these individuals. We usually do that in the form of this drug called L-DOPA. However, big negative consequence of that is that it turns out that providing this drug is actually excitotoxic. So by providing this dopamine replacement to hopefully replace some of these degenerated signals, you're actually going to cause damage to the caudate and putamen, to the corpus striatum. This means that this is very much not a permanent solution. And although providing L-DOPA to a patient can reduce symptoms in the short term, it can actually increase the speed at which Parkinson's develops. Another strategy has been deep brain stimulation. And in particular, we see deep brain stimulation to the subthalamic nucleus being quite useful. So, what I would suggest to you is that you refer back to your notes and even previous slides in this particular lecture and think about how stimulating the subthalamic nucleus would affect voluntary movements. We can basically go back to a slide like this. If you were to stimulate the subthalamic nucleus, that is going to activate this globus pallidus, which is going to inhibit the VAVL complex, and therefore it's going to inhibit activity in the frontal cortex. So the idea would be that it's actually going to inhibit the undesired movements. So this is actually something we can see. This is, there we go, deep brain stimulation. This is something that's actually used to help prevent some of those undesired movements, such as maybe the tremors. I would recommend maybe watching this real life demonstration video. It is pretty extraordinary. It's a man who has one of these deep brain stimulators. It looks like he's pretty advanced in his Parkinson's. Uh, when the stimulator is on, he is completely still. When it's turned off, he immediately just starts shaking. His entire body experiences tremors. So this type of thing can be quite useful in inhibiting those undesired movements. Remember, that's what the indirect pathway is mainly connected to. It's those undesired movements that are sort of conflicting with the voluntary movements that you desire. So, deep brain stimulation and L-DOPA are two possible ways of helping to alleviate at least some of the manifestations of Parkinson's disease. And the next video will take a look at a different basal ganglia disease, which is Huntington's.